Now, from the detection of designer drugs, our next speaker will be presenting the development of vaccine as a possible opioid therapy. I would like to introduce our next speaker. I'll just share my slide. Okay. Our next speaker is Dr. Rodell Barrientos. And Rodell joined the US military HIV research program and the Henry M. Jackson Foundation for the Advancement of Military Medicine at the Walter Reed Army Institute of Research as a postdoctoral fellow in August 2019. Dr. Barrientos currently works in the laboratory of Dr. Gary Matias, where he develops immunoconjugate vaccines against heroin and fentanyl and applies mass spec approaches for vaccine characterization, quantification of drugs of abuse in biological samples, and antibody-antigen interactions. Rodell obtained his PhD in medicinal biochemistry from the University of North Carolina, Greensboro, where he developed novel and advanced methods to analyze complex glycolipids using liquid chromatography and mass spectrometry. He was also a recipient of Graduate Student Award from the American Society for Mass Spectrometry in 2018, and he is currently the program chair and vice president of the Washington Chromatography Discussion Group. He is an elected full member of Sigma Psi Scientific Research Honor Society, and currently he is a member of the PAASE. Let us all welcome Dr. Rodell Barrientos.
Good morning to all of you in the Philippines and to us here in the US. Good evening. I'm really delighted to be here today. And thank you very much, Dr. Completo and the rest of the organizers and the PAASE for the invitation and for organizing this event. I know it's a lot of work and well, we really appreciate it. So let me share my screen. Okay. Okay, so I hope everyone is able to see my screen now. So before I begin, these are my disclosures. So first, I am presenting my own views, which do not necessarily reflect the views of the US Army, the Department of Defense, or the NIH. Number two, all animal experiments complied with the Animal Welfare Act, and all protocols have been approved by our IACUC. And number three, that all other authors of the work are inventors of a provisional patent filed by the Henry M. Jackson Foundation for the Advancement of Military Medicine. So the opioid epidemic has claimed thousands of lives in the last 2 decades, and the current COVID-19 pandemic further worsens the situation. On average, 128 people die each day because of opioid overdose. And this graph shows the number of deaths due to heroin. And we can see that it's to decline beginning 2014. So alarmingly, heroin is being adulterated with more potent opioids such as fentanyl and called street heroin. Deaths due to adulterated heroin has increased steadily and about to outnumber the deaths due to heroin alone. And most victims unknowingly consume the deadly fentanyl. So fentanyl is being added to heroin to lower the dose needed to achieve euphoria and to let the drug dealers earn more. So in the body, opioids act on opioid receptors. So opioid receptors are widely distributed, but they are most abundant in the brain and the digestive tract. But most of its effects are exerted in the brain. So there, the opioids bind to opioid receptors, which results in the reduction of pain, euphoria, constipation, and the euphoria, also called the high, or the state where your brain thinks that everything is going very well, and this is what makes opioids addictive. So one serious side effect of opioids is the depression in respiratory and heart rate. So as Dr. Herona presented earlier. So opioids could lead to sudden stop in breathing and resulting in death. And in fact, deaths due to opioids are mainly caused by respiratory depression. So for opioid dependents who undergo opioid use disorder treatment, such as the use of methadone, buprenorphine, naltrexone, and naloxone, there is actually higher risk of overdose when they relapse. So it is thus important to develop strategies to protect these vulnerable populations. So to address this, our approach being an immunology lab is to develop vaccines. So the idea is simple. So as you can see here on the left side, so in an unvaccinated human, when opioids get into the blood, okay, it will travel to the brain, cross the blood-brain barrier, bind to opioid receptors, and there it induces its effects. Vaccines are designed to inhibit this process, as you can see on the right side. So vaccines will induce antibodies that would sequester the opioids in the blood. And because the drug antibody pair is too large, it will not cross the blood-brain barrier, therefore preventing its access to opioid receptors in the brain and thus blocking its physiological effects. So what are we looking for in a good vaccine? So among the desirable traits of the vaccine include induction of high titer, high affinity, and durable antibodies. It is also important that these antibodies don't bind to the drugs used for opioid abuse therapy. So, and the vaccine should be able to protect experimental animal models against opioid challenge. So let me give you a brief overview of the biochemistry of heroin. So heroin is basically metabolized in the body in multiple steps. So the first steps can occur in the blood via the action of esterases. So as you can see, it has two ester functional groups. So the esterases act on these ester groups to generate the metabolite 6-AM or 6-acetylmorphine and morphine. And the heroin's addictive effects have been attributed to heroin as well as to these two metabolites. Further metabolism in the liver produces glycosylated metabolites, which do not have any addictive effects. So thus, a good vaccine against heroin should be able to induce antibodies against heroin itself, 6-acetylmorphine, and morphine. So as Dr. Herona mentioned about the danger of fentanyl, so let me further talk on that in the context of our vaccine. So fentanyl is the most commonly found adulterant in street heroin that cause many fatalities. It is a synthetic drug and its structure is shown here. So the strength or potency of fentanyl can be modified by altering the chemical structure on these crucial sites as shown by the red arrows on the fentanyl chemical structure. For example, when we introduce an ester group in the piperidine ring, okay, the potency of the drug can be increased. And now, based on the latest list that I have searched, there are at least 40 known analogues of fentanyl. So in fact, this picture from Drug Enforcement Administration reflects how serious fentanyl analogues are. Compared with heroin, as you can see from the picture, extremely small amount of fentanyl and carfentanil can cause death in humans. Importantly, it's very easy to be exposed to fentanyl because it can be through inhalation, ingestion, injection, or even skin contact. So small molecules generally don't generate immune response. And that's why vaccines to drugs of abuse are formulated differently. So here, the vaccine is composed of a hapten, okay, a carrier protein. In this case, we are using tetanus toxoid, and an adjuvant. And in this case, we are using an army liposome formulation developed by army scientists in our institute. So to make them into a vaccine, opioids need to be attached to a protein. And therefore, surrogate molecules called haptens that resemble the target opioids need to be synthesized. So we previously reported a heroin vaccine, and this vaccine uses a hapten called 6-AMHAP, as you can see on your screen, which our group published in 2018. And this is now being slated to undergo phase 1 clinical trial very soon. So could that vaccine work against fentanyl? The answer is no, because heroin and fentanyl are structurally distinct. And so antibodies developed against the heroin vaccine may not recognize the fentanyl and fentanyl analogues and vice versa. So now, together with our collaborators at NIDA, or the National Institute on Drug Abuse at NIH, we developed a hapten called PAASE, for the fentanyl vaccine, which we just submitted for publication. And I'm actually happy to share with you that this morning it got accepted in molecular pharmaceutics. So I wasn't able to revise the slides, and that was a very good news. So in our vaccine, haptens are attached to tetanus toxoid to form the immunogen, and added with the army liposome formulation, which is an adjuvant. And then adjuvant is added in order to, as an immunostimulant to enhance the immune response against our immunogen. So the chemistry for the production of the immunogen is very simple. First, we incubated the carrier protein. So the carrier protein contains surface lysine groups, which are free primary amine groups. And afterwards, the tetanus toxoid linker was cleaned up to remove the excess linker. So the linker that we're using is called SMPEG2 linker. So it is a hetero-bifunctional linker. So one end of it reacts with the primary amine, and then the other one is a maleamide group that reacts with free thiol. I'm sorry. Can somebody please make sure that they are muted? All right, thank you so much. So yeah, as I was saying, so the linker is hetero-bifunctional, and one end reacts with the primary amine on the surface of the carrier protein, and the other end reacts with a thiol group. So once we generate the TT linker, or the protein conjugated with the linker, we then reacted the hapten that contains a free thiol group that reacts with the maleamide in the linker. So to generate the immunogen, either the TT-para-AMFENHAP in case of fentanyl vaccine, or TT6-AMHAP in case of the heroin vaccine. So we are using MALDI-TOF mass spectrometry to measure the resulting molecular weights of the conjugates. And from the shifts in masses, as you can see on the right side, I have shown a representative MALDI-TOF mass spectrum, wherein you can see the mass of the unconjugated tetanus toxoid and the conjugated tetanus toxoid with the heroin hapten. And from the shift in mass, we can calculate the number of hapten molecules that are attached to the tetanus toxoid, and that is called hapten density. So hapten density is a very important measure of quality, as well as it relates to the efficacy of the vaccine. So consistently, we found about 30 to 35 haptens per carrier protein molecule. So to address the epidemic of overdose due to fentanyl adulterated heroin, we developed a combination vaccine consisted of the TT6-AMHAP or the heroin vaccine and the TT-para-AMPHENHAP or the fentanyl vaccine. So the goal here is very simple. So we want to induce dual immunological response that would elicit two populations of antibodies, one population recognizing heroin and its metabolites, and another population recognizing fentanyl and its analogues. So to determine if the vaccine is effective, we initially tested it in mice. So we are using female bulb C mice for these experiments. So we injected 50 microliters of the vaccine, and the composition of the 50 microliter is shown on the slides. So we vaccinated the mice at weeks 0, 3, 6, and 14. And we collected the blood at indicated weeks for the measurement of antibodies using ELISA. Then at week 18, we challenged the animals with opioids. So the graphs shown here are the results of ELISA measurements. So it is reported in terms of antibody titers. The black bar represents unimmunized mice, and the blue represents the immunized mice. So we can see that for those mice that did not receive the vaccine, their blood didn't show antibodies that bind to the hapten. Whereas those mice that received the vaccine generated antibodies that are recognizing the fentanyl hapten as well as the heroin hapten. So these results suggest that the vaccine is immunogenic in mice and can induce antibodies against the hapten. So since our goal, as I mentioned earlier, was to induce antibodies that can bind the drugs in the blood and prevent access to the brain, we then asked whether the serum from vaccinated mice could actually sequester the opioids in vitro. So to do this, we use equilibrium dialysis, and this method uses a plate that is commercially available that contains a sample and buffer chamber that is separated by a semi-permeable membrane with a 12 kilodalton cutoff. So this cutoff is too small to allow proteins to partition. And therefore, if the drug is bound to antibody, it becomes too large that it would stay in the sample side. So how the assay works is simple. So the serum is incubated with the drug of interest and in the sample chamber against a buffer. Normally, we're using the phosphate buffered saline or PBS, and we incubate it for 24 hours at 4 degrees C. So after the incubation, of course, some of the drugs will partition into the buffer chamber. Okay, so the concentration of the drugs present in both chambers are measured by LC-MS-MS, and the fraction bound can be calculated using this equation. So this method has been published in 2016 from our lab. So shown here is the dilution binding curves of sera from vaccinated mice. So the y-axis is the fraction bound, and the x-axis is the dilution of serum used for each assay. So we can appreciate that the week 0 sera, okay, so which is the serum collected before we injected the vaccine, okay, the blood did not bind the heroin, fentanyl, 6-acetylmorphine, or morphine, okay? But the week 16 sera, okay, bound these opioids even at very high serum dilutions. So this suggests that vaccination of mice successfully induce antibodies that can sequester the target opioids in vitro. So we also investigated the average antibody affinity using a method based on liquid chromatography tandem mass spectrometry that is published in our lab. So the antibody affinity was measured in terms of the KD, as you can see on this cartoon representation here. Okay, so the KD is the dissociation constant, and the KD measures the strength of the antibody-antigen interaction. So the lower the KD, the stronger is the interaction. So we found that the KD values are in the nanomolar and subnanomolar range for all the drugs tested in vitro. And this tells us that once the drug binds to antibodies, it will take longer time for it to dissociate, and thus less likely to cross the blood-brain barrier. Or in other words, it could actually totally prevent the access of the drug to the brain or slow it down to the point that it will not be very efficacious. So next we asked whether the antibodies generated can actually sequester fentanyl analogues. So this is very important because we want to have a vaccine that is effective not only for fentanyl, but also to fentanyl analogues. So here, we use 5 of the most common analogues involved in overdose deaths. So we observe that these drugs were effectively sequestered by post-immune sera, that is week 16. So we have the cyclopropyl fentanyl, furanyl fentanyl, cis-3-methyl fentanyl, parafluorofentanyl, and carfentanil. We can also see that the antibodies generated by the mice had varying strengths of binding to these analogs, but nevertheless, all of these analogs have been bound successfully by the generated antibodies in the blood of mice. So overall, these data tell us that the vaccine can effectively induce antibodies that can sequester not only fentanyl, but also its analogs, at least in vitro. So importantly, as I have mentioned earlier, the antibodies should not cross-react with drugs that are currently used for treatment. So therefore, we asked whether the serum antibodies can cross-react with drugs used for opioid management therapy. So we found that none of the drugs, methadone, naloxone, buprenorphine, and naltrexone, was significantly sequestered by post-immune sera, highlighting that vaccination will not jeopardize the therapeutic use of these drugs, and as such can be used as complementary therapeutics to these existing treatments. So now, what I have just mentioned is in vitro assay, right? So we really want to see if this is effective in vivo. So to see if the vaccine is effective in vivo in protecting mice against opioids, mice were injected with an increasing dose of an opioid on week 18. So we gave the opioids subcutaneous injection. Okay, so we know that opioids can block pain. Therefore, we use the transient pain assay, hot plate test, to evaluate vaccine efficacy. So in this test, before and after injecting the opioid, the mouse was placed on a hot plate at 54 degrees Celsius. So the time it took for the mouse to feel pain, such as jumping or leaking, was recorded. And 12 seconds was used as a cutoff time to prevent injury on the mice. So the recorded times were then converted to percent MPE following this equation shown on the upper right side of the screen. So to easily interpret the succeeding slides that I'm gonna show you, just remember that a high percent MPE means that the opioids get into the brain, giving no pain sensation to the mice, or at least very little pain sensation. But if the MPE is low, it means that the mice feel the pain, and therefore the opioid isn't able to cross the blood-brain barrier. So shown here is the plot of the heroin challenge experiment. So the x-axis is the cumulative dose, and the y-axis is the animal response. So we can see that in the hot plate test, the unvaccinated mice, okay, achieve 100% MPE. So the black line, okay, achieve 100% MPE at a relatively lower dose, okay? The vaccinated mice, on the other hand, which is the yellow and the blue color lines, achieve 100% MPE at a relatively higher dose. So which means that we are effectively shifting the dose response curve and giving us an ED50 shift of about 3.5. And what does this tell us? This tells us that the potency of heroin was decreased significantly by vaccination. So we can also appreciate that combining the fentanyl vaccine with the heroin vaccine had no effect on the efficacy, therefore suggesting that combination of two immunogens is actually a feasible approach in drugs of abuse vaccine development. So this tells us overall that immunization attenuates the effect of heroin in vivo, and that immunization can be used to blunt heroin-induced effects. So next is that we also challenge the animals with fentanyl, okay? So we can see that the unvaccinated mice, also in black color here on the graph, reach 100% MPE at relatively low fentanyl dose, whereas the vaccinated group achieve 100% MPE at a much higher dose, effectively shifting the dose response curve to the right by approximately 6.7-fold in terms of the ED50. So this tells us that the potency of fentanyl has been decreased or blunted by vaccination. So we can also appreciate that, you know, the combination vaccine, the presence of heroin vaccine in the formulation did not affect the efficacy. So this suggests that the vaccination attenuates the effect of fentanyl in vivo, and that immunization can be used to blunt fentanyl effects in mice. So finally, we challenged the mice with a combination of heroin-fentanyl, 10-to-1 in terms of weight ratio. So the graph, the black represents the unvaccinated mice, and we can see that the unvaccinated mice reach 100% MPE at relatively low dose compared to the vaccinated group. So there is a shift in the dose response curve of about 3.4-fold in terms of ED50. So this suggests that the vaccine was able to attenuate the potency of combination heroin-fentanyl in mice. So in summary, we developed an immunogenic and efficacious combination heroin-fentanyl vaccine that protected mice from opioid-induced effects, particularly of heroin, fentanyl, and combination heroin-fentanyl. The vaccine was able to induce antibodies with high titers and high affinity that was able to bound fentanyl analogues, heroin, and its metabolites, but did not bind drugs that are used for opioid use disorder therapy, and that the combination fentanyl heroin vaccine is actually feasible, at least in our preliminary studies in mice. So despite this data, there's still a lot of exciting work to do. We are planning to determine the pharmacokinetic and immune correlates of protection, which will utilize a lot of method development on the mass spectrometry side. We also plan to do studies in rats, and we are currently developing monoclonal antibodies for these drugs and use them to actually perform passive immunization experiments and to see whether we achieve the same level of protection that we are seeing with active immunization with our vaccine. So with that, I'd like to thank Dr. Gary Matias, my principal investigator, and Dr. Oscar Torres for letting me work on this project. So the members of the adjuvants and formulations section at the Walter Reed Army Institute of Research, especially Connor and David, who performed some of the animal experiments. Our collaborators at the National Institutes on Drug Abuse under the Kenner Rice Lab, and MHRP leadership for supporting this work. This work was funded by the National Institutes of Health through its HEAL, or the Helping to End Addiction Long-Term Initiative, and the project was supported by the Henry M. Jackson Foundation for the Advancement of Military Medicine. So with that, I will be happy to take any questions.
Thank you very much, Rodell, for that very interesting talk. We are now open to questions from the audience.
I think there's a question from Michelle Autentico. She raised her hand earlier. Michelle? Michelle Autentico.
You may have inadvertently clicked on the raise hand button.
Maybe.
Yeah, I think it happens.
Yeah.
There's another question here from Ronilo Flores. So we would like to ask about your opinion on the administration of the putative vaccine on cancer patients who use fentanyl patches or other patients under legitimate fentanyl prescription. Have you tried producing monoclonal antibodies?
Okay, so that I think is a very important question to address because the fentanyl is a Schedule II drug in the US, and I am not familiar how it is scheduled in the Philippines, but actually in the US it's Schedule II because it has medicinal or medical applications. And if you are vaccinated, assuming that this vaccine goes into humans, and you are vaccinated with the fentanyl vaccine that I talk about. So what happens is your body generates antibodies against these substances. Therefore, in the future, if you're gonna need fentanyl for, let's say, pain medication treatment, or maybe for palliative treatment, right? So the fentanyl will not be effective to you anymore. And maybe it will be more, you need to find an alternative to fentanyl in that respect. So probably morphine. But if you are injected with a combination heroin-fentanyl vaccine, there could be a problem with morphine. But that's why we are tweaking a lot of chemistry here in the development of these vaccines in order to make sure that the antibodies generated by vaccine is highly selective to the target opioids and not to other painkillers that could be used in the future by physicians. So the second question is, have you tried producing monoclonal antibodies? We have produced monoclonal antibodies, but we haven't published it yet. So I haven't included it in my presentation today. But we are developing monoclonal antibodies against fentanyl, fentanyl analogues, as well as heroin. So it's ongoing. We have both mice, we also have rats, and we also have humanized monoclonal antibodies that are currently under development.
So there's a question from YouTube. It's from Connie Remorosa. Nice work, Rodell. Her question is, could you elaborate a little bit more regarding the binding quantity, KD, by chromatographic technique?
Hi, Connie. How are you? Thanks for the question. So basically, the KD measurement uses equilibrium dialysis. So in equilibrium dialysis, what we can do is incubate an unlabeled drug. So we are using two versions of the drug in the UPLC-MS-MS measurement of KD, okay? So the unlabeled drug is first incubated with the serum, okay? And then on the buffer side, we actually have a varying concentration of the labeled version of the drug. So the idea is we let the labeled drug to compete with the unlabeled drug in the process. So during the competition, it's like competition ELISA. So we can measure IC50. So from the IC50, we can actually use an equation developed by Mueller in the 1980s, utilizing radioactive versions of the method. But in our case, we're using mass spec. So from that equation, we can calculate the value of KD. I can share with you the methodology if you are interested, the actual details of the steps.
Hi, Rodell. Thanks for the interesting talk. I have a question about metabolites because a lot of the fentanyls are actually rapidly metabolized in the human body. So have you looked at affinities for the potential metabolites of fentanyls using some of the vaccines that you're developing?
Oh, thank you so much for that very insightful question. Actually, we have not looked at fentanyl metabolites yet. The only one that I have looked at is actually norfentanyl. So that is the most common core metabolites of fentanyl analogues. And not unexpectedly, the antibodies generated in serum successfully strongly binds the norfentanyl. But yeah, there's still a lot of things we need to work on here. And one of those is the metabolites. And at the same time, we plan to expound the affinity measurements to other fentanyl analogues. Because right now, we have limited access to fentanyl analogues. So we just got renewed for our DEA license, so maybe we can purchase more. So from there, we can start measuring the other analogues.
And you're actually going to be doing some studies on the pharmacokinetics of the process, right? Because then a potential complication would be what is the relative elimination half-life of the drug versus how fast it can be captured by the antibody in the human body?
Yeah, I think it would actually depend on how the drug was administered. For example, in our case, in the mice model. So most of the time we are measuring through subcutaneous challenges, but we are also doing IV challenges. And we have that in IV challenges, of course, the kinetics is faster, and therefore the elimination half-life is also faster. And even though it's IV and the kinetics is fast, we were able to get comparable efficacy of the vaccine. So we believe that, maybe in terms of inhalation, we haven't tested that. Probably it would take shorter time for the drug to reach the brain, and therefore the antibody might not be able to capture that very quickly. But at least in subcutaneous and IV, the antibody works very well. But yeah, we have limitations right now in terms of the challenges that we do in the animals. So maybe we need to try other routes. Thank you for the for that question.
Thanks for the insightful answer.
Hi, Rodell.
Hello.
Yeah.
Go ahead, Danny.
Yeah. There's another question from the Q&A notes on Zoom.
Okay.
Have you considered using other hetero bifunctional linkers aside from the SMPEG linker?
Okay, wow, that's a very very nice question on the chemistry side. So actually, the selection of the SMPEG2 linker it has been informed by numerous conjugation studies, not necessarily from our lab, but from the literature. So the reason why we chose SMPEG2 is first, the presence of PEG2 or the polyethylene glycol unit in the linker has been known to be, relatively stable in humans, and also, although it could generate also antibodies against the linker, the presence of pre-existing immunity against the linker actually do not have a negative effect on the efficacy of drugs and other pegylated molecules that have been reported in the literature. So that's the primary reason that we use that linker. I hope I was able to answer the question.
Thanks, Rodell. There's a lot of questions on the feed about COVID-19. Do you want to just give your general thoughts about the perfect drug and the perfect vaccine for COVID-19? We don't have a lot of time, but just to try to address a lot of these. We've got about probably 6 to 10 questions that are similar in nature on COVID-19. Would you like to give your thoughts?
Yeah, actually, our lab is involved in the development of a COVID-19 vaccine. Our lab particularly is the one that is producing the adjuvant for the Army COVID-19 vaccine. It's called the SP-Fen vaccine. So basically, what I can say about this, I am not fully aware of this current vaccine that's being developed. But what I'm thinking is there's a lot of approaches that we can go to. For example, so the mRNA vaccine that is progressing to phase 3 clinical trial, and that is manufactured by Moderna in the US. So that's one promising approach. Another promising approach is through the use of recombinant proteins, and that's what the vaccine being developed in our institute is. So it is based on the spike protein of the virus, and that is added with an adjuvant, and it would generate antibodies against the spike protein on the virus surface that would prevent its binding to the ACE2 receptor in the host cell, and therefore it will prevent the infection. So I have limited information on this, but my take is I think mRNA is a very promising vaccine approach. It's novel and it's first in human, and we'll see. And the earlier results of these vaccines are really promising, but we need to wait until we reach into phase 3 that we can actually conclude whether this is really efficacious in the real world scenarios.
Thanks, Rodell.
So, Rodell, I have a follow-up question about your recombinant protein that's being done in your lab. So I'm assuming your recombinant protein is still glycosylated, or it's not glycosylated anymore? Because it's important, right? Because of the glycan shield.
Yes. I am not aware whether the recombinant protein that is being used is actually glycosylated, and that's a very good question. Yeah, because as I have mentioned, our lab is involved in the adjuvant formulation, And another lab, which is the Gordon's lab, is the one that is producing the protein antigen. But I suspect because through the recombination experiments, it should be able to, you know, have glycosylation on the protein, right? So it's just I'm not truly aware whether the final antigen to be used in the vaccine contains these glycosylations.
Okay, thank you.
All right.
So I have another question, maybe last question. So you mentioned that your vaccine can distinguish between the heroin and then morphine and the simpler analogs, right? So have you identified the pharmacophore where your vaccine is recognizing on your heroin or morphine or your fentanyl?
Yes. I haven't included that in my presentation, but I'm writing a paper right now, actually on the facial recognition of the antibodies, because I have a slide. I don't know if I can go back, but if you remember the serum binding experiments, I use different fentanyl analogues, and the binding curves have different shapes, right? So which means that these antibodies recognize a particular portion of the molecule or the pharmacophore that binds to the paratope or the antigen binding site in the antibody. So we have a hypothesis on that, and we are still confirming it, and hopefully it will be a paper soon. So yeah, so we believe that in the fentanyl structure, the piperidine ring plays a crucial role, as well as the let me try to go back. So maybe, okay, here. As you can see here, can you still see my slides, or am I still sharing it?
Yes, you are.
Okay, so as you can see from the chemical structures here, okay, so when you replace the phenyl ring on this amide portion of the molecule by cyclopropyl ring or furanyl or maybe an acetyl group, actually, or let's say, let's focus on these two first. So the binding doesn't show like much difference, but if you replace that portion with acetyl group and also add a methyl group on the piperidine ring, suddenly there is a change in the binding profile. So this tells us that the piperidine ring plays an important role in the binding of the drug with the antibody, but these moieties on the amide portion of the molecule play only small role, right, on the antibody recognition. And as you can see here on the carfentanil, this is further supported by carfentanil data. See, if there is an ester group on the piperidine ring, the bind is weaker. So, so that's actually our hypothesis, that the piperidine ring and whatever substituent on the amide portion of the molecule is actually tweaking the antibody recognition. So yeah. We just need to do like maybe X-ray crystallography studies. So we are actually looking for collaborators to do that for us.
Okay, thank you.
So I think that's the last question that will be addressed. And now we will move on to the last speaker.
Thank you so much.
Thank you very much, Rodell, for that very interesting talk. And then I would like to call on Dr. Isagani Padulina to introduce our last speaker, Dr. Michelle Regolasio.
Okay.